Hello student, now we are going to discuss hemostasis or clot formation, mechanism of stopping of bleeding. So whenever there is any cut of your blood vessels in the body, we know that there are certain mechanism by which the bleeding will be stopped from that place, and that is the process of hemostasis, right? So hemostasis, how it happens, right? Whenever there is any injury to the blood vessel, the first thing that is going to happen is vasoconstriction. And that vasoconstriction will be transient. So, I am writing here, first there will be transient vasoconstriction. This transient vasoconstriction is mediated by serotonin, right? After the transient vasoconstriction, it will try to decrease the bleeding a little bit, but it will not be able to completely stop the bleeding. After that, what is going to happen? There will be formation of primary hemostatic plug formation will be there. And this primary hemostatic plug formation is mainly done by the platelet, by a mechanism, I will tell you. And after the primary hemostatic plug formation, the clotting system, the fibrinogen mediated clotting system will be activated, and that will lead to definitive plug, okay, which is nothing but clot formation. So whenever the clot is formed, the bleeding will stop completely. So this way, whenever there is any injury to the blood vessels, the body is going to stop the bleeding by this mechanism. Now, first of all, we are going to understand how this primary plug formation is going to happen. So, primary platelet plug formation, right? It happens in 3 stages. Number 1, adhesion of platelet. Number 2, activation of platelet. And number 3, aggregation of platelet. By this 3 stage, what will happen? The area where the vessel is injured, that place will be plugged by the platelet initially. This is known as the primary platelet plug formation. So let's see how this happens with the help of a diagram. So suppose this is your vessel wall. Okay, suppose this is your vessel wall, and the endothelium cells are there, right? Normally, platelet will never adhere on these endothelium cells, because platelet also contain negative charge, endothelium contain also negative charge, and there is a coating, coating on this endothelium cell, because of which platelet never adhere here. But suppose there is an injury here, okay, and there is a disruption of this endothelial cell in this region. What will happen whenever this kind of injury happens? The subendothelial collagen will be exposed. So suppose this is subendothelial collagen, it is exposed. Not only that, subendothelial connective tissue also contain von Willebrand factor. I hope all of you know that von Willebrand factor, it is synthesized by this endothelium cell also. It is present inside the endothelium cell as an ovale pallid body. It also present at the subendothelial connective tissue, and it is also secreted by platelet macrophage cell. Platelet macrophage cell, right? So von Willebrand factor and collagen is exposed. Now, in this area, platelet will adhere. Okay, so this is known as the adhesion step of platelet. So this is platelet. How it is going to adhere? There are receptors on the surface of platelet. One receptor is, which is interacting with this von Willebrand factor. This is a glycoprotein GP1B1X9. GP1B9 receptor, right? So this is glycoprotein receptor which is present on the surface of the platelet, 1B9 receptor. It is going to interact with von Willebrand factor. So this is the first attachment point, von Willebrand factor and GP1B1X. There are other receptors also on the surface of the platelets that we know. This is known as glycoprotein 2B3A. What is the role of this receptor? I will tell you a little later. But now look at GP1B is there, GP2B is there, GP3A is there. So if I ask you where is GP1 and GP2A, 1A and 2A. So GP1A and 2A, GP1A and 2A, they interact with this collagen. 2A and 1A, they interact with this collagen molecule which is exposed here. Now, because of this interaction of platelet, right, with the collagen and von Willebrand factor, certain intracellular signal will be produced. Okay, we know like a cell which is having its receptor. So whenever hormone binds with the receptor, what happens? The second messenger system will be activated. In the same way, inside the platelet, there will be activation of certain cascade system, due to which it will produce IP3-DAG second messenger, and that will lead to influx of huge amount of calcium inside the platelet. Now, whenever calcium is present hugely inside the platelet, there will be certain changes which is happening within the platelet, that is known as activation stage of platelet. So after this adhesion, so I can understand whenever the platelet is adhering here, this is the adhesion stage, the receptor on the platelet surface is activated, that is generating certain signal which is producing calcium. In presence of calcium, certain changes is happening inside the platelet. What are the changes? There will be change in shape of the platelet. Platelet will change its shape. Not only that, there will be degranulation of platelet. There will be degranulation of the platelet. We have already seen there are different types of granules in the platelet, alpha granules and dense granules, and they will secrete their component, like for example thrombin. Okay, thrombin will be released, thromboxane A2. Okay, not only that, platelet activating factor, all of this factor will be secreted due to this degranulation. And not only that, the third one is changes in conformation of GP2B3A receptor will change its conformation change. So these three things is happening, and this is known as the activation stage of platelets. So change in shape, degranulation, and GP2B3A receptor is changing its conformation. Now, whenever it is secreting this thromboxane A2 platelet activating factor thrombin, it will act on the nearby platelet which are present here. And whenever it is acting on the nearby platelet, you know, the other platelet which are present in the nearby area, they are also going to be activated. Okay, they are also going to be activated. So suppose this is another platelet, it is active, and this is another green color platelet, this is also active. So whenever this green color platelet are also active, the receptor which are present on their surface, that is GP2B3A, it will also be activated. Here also the GP2B3A receptor will be activated. Now this GP2B3A receptor is active on one platelet, and another GP2B3 receptor is active here. In between these two receptors, fibrin molecule will bridge. Fibrin molecule is bridging in between these two receptors. By this way, what happens? One platelet will aggregate with other platelet with the help of this fibrin and GP2B3 receptor. This step, when all the platelets are coming close to one another and they are forming an aggregate, this is known as aggregation step of platelet plug formation. So whenever the platelet is forming an aggregate, the area where it was damaged, this area is going to be sealed by platelet, and this is known as primary platelet plug formation. So what are the steps? Addition of platelet, activation of platelet, and aggregation of platelet. Now, after the platelet plug formation, on the surface of this platelet membrane, the coagulation cascades will be activated, and that is known as definitive clot formation or definitive plug. So on the surface of platelet membrane, the coagulation pathway will be activated. Now this coagulation pathway, we all know that there is one intrinsic pathway and there is one extrinsic pathway. Look carefully, this is important. So this is intrinsic pathway of coagulation and this is extrinsic pathway of coagulation. Okay. Sometimes it is known as secondary hemostatic plug or definitive clot formation. Anyway, whenever you keep blood inside the test tube, suppose this is a test tube and you have kept blood here without any anticoagulant, we know the blood will form a clot. Which pathway of coagulation is activated here? This is the intrinsic pathway, contact pathway. This glass surface contains negative charge. In presence of this negative charge, the clotting system will be activated and the clot will form inside the test tube. But you know, between these two pathways, in vivo, inside your body, whenever vessel wall is injured, clot formation is initiated by extrinsic pathway. This is very, very important. If you look at the physiology textbook, particularly Guyton, you are not going to get it. But if you open up Harrison and other research article, it has been proved now that it is not the intrinsic pathway which is activated first time in vivo. In vivo, the most important pathway which is initiating your coagulation cascade, which is starting your coagulation cascade, that is the extrinsic pathway. So this is the start point. This is the starting point of coagulation. So what happens whenever there is injury to the blood vessel? I have told you already that collagen will be exposed. Not only that, the subendothelial connective tissue, they are going to release certain factor, and one of such factor is factor 3, which is also known as tissue factor. So whenever this tissue factor is released from the subendothelial connective tissue, this tissue factor is going to combine with factor number 7, and this tissue factor factor 7 combination is going to do two important jobs. It is directly activating the factor 10 as well as it is directly activating the factor 9. Getting my point? So initiation of the coagulation system is starting from this extrinsic pathway and it is done by tissue factor. That's why this is known as the tissue factor pathway also. Okay. Tissue factor pathway also. Tissue factor is releasing, combining with factor 7. Whenever they are forming a combination, they can directly activate factor 9 as well as factor 10. Now, we all know that if the factor 9 is activated with the help of factor 8, it will ultimately activate this factor 10 to active factor 10. Or even suppose this pathway is also not there, still also this factor 7 and tissue factor combination has directly activated the factor 10, right? After this activation, the tissue factor pathway will shut down. It will shut down. How it will be shut down? It will be shut down by a protein which is known as tissue factor pathway inhibitor. So, it is just initiating the coagulation cascade. After initiation, it will shut down itself. Getting my point? So, it is acting initially. It is starting the pathway, but after starting, it is stopped. Right. So, Whenever you have already initiated the activation of factor 10 and factor 9, now your coagulation system is not going to stop. Why? Whenever you activate this factor 10, look at this pathway. What will happen? The downstream cascades will start. That is, active factor 10 is converting prothrombin to thrombin, and then thrombin will convert fibrinogen to fibrin, and fibrin means this is nothing but your clot. But how this clot formation will amplify? Because after initiation you told that this pathway is shut down. So who will maintain this pathway? Who will amplify this clot? Huge clot means you are going to block the vessels. Otherwise the clot is not sufficient to block the vessels. So remember, this thrombin is very, very important. Whenever little bit of thrombin is generated in your blood, a very little amount of thrombin, it can directly activate two important clotting factors. It can directly activate factor 5 as well as it can directly activate factor 8. Very, very important. Right. So, extrinsic pathway has just started activation of the coagulation cascade, and it has activated factor 10 and factor 9. Whenever factor 10 is activated, I have generated a little amount of thrombin, and that thrombin is generating a positive feedback system. Why? This thrombin is stimulating factor 5. Factor 5 can activate factor 10. This thrombin is stimulating factor 8. Factor 8 can activate factor 9, and then it will activate factor 10. So now I can understand that whenever you generate a little amount of thrombin, now the coagulation pathway will move like this, will move in this direction only, will move here only, here only, here only, here only. And whenever the pathway is moving in this fashion, what you are generating? You are generating more and more amount of thrombin. And whenever your thrombin generation is huge, ultimately this fibrinogen will form fibrin and clot formation will be amplified. That's why we say that coagulation cascade is a positive feedback system, and this positive feedback system will set in whenever little amount of thrombin is generated. So you can understand that from this to this region, this is the main thing which is important in coagulation pathway. These are the main factor which is important for amplifying the clot. So what about other factor? Tell me, what about the role of this other factor? Do you think these factors are very, very important? Answer is no, no, no. That's why Harrison is saying, if there is deficiency of this factor 12, even if there is deficiency of this factor 11, clinical bleeding is not going to happen. But you look at, if there is deficiency of this factor 8, or if there is deficiency of the factor 9, they will lead to hemophilia, right? Because you have already seen that this is the area where positive feedback system is operating, and this is amplifying your clot. And that's why we have to remember that the importance of this intrinsic pathway activation in vivo for in vivo coagulation system, it's not very important. Main important is the extrinsic pathway which will initiate, and then the thrombin will generate the positive feedback system and clot formation will be done, right? Now, after this discussion of this coagulation pathway, you know that there are certain coagulation system pathway checking investigation by which you can check the coagulation profile of the subject, and two important testing are done, that is the PT, prothrombin time, and APTT, activated partial thromboplastin time. So APTT and PT, prothrombin time. If your prothrombin time is more than normal, then think of the extrinsic pathway factor only. If your APTT is prolonged, you think of deficiency of the intrinsic pathway factor. So look at again. So you look at the color coding. If you look at the color coding, so what you're finding out, This yellow color factor which I have written here, this is the common pathway. So from factor 10 onward, this is known as the common pathway. And this is the extrinsic pathway, and this is the factor of intrinsic pathway. If there is any deficiency of factor in the extrinsic pathway, then your prothrombin time will be prolonged. If there is deficiency of any factor in the intrinsic pathway, then your APTT will be prolonged. If there is deficiency of any factor in the common pathway, both PT and APTT will be prolonged, right? Now, suppose I have a deficiency of factor 12. My APTT will be prolonged, but am I going to suffer from significant clinical bleeding? Answer is no. And the explanation is already given. So that's why if you look at this coagulation test information which is given in Harrison, prolonged PT, prothrombin time, means deficiency of factor 7. Although vitamin K deficiency can also causes increased prothrombin time, because vitamin K dependent clotting factor, all of you know, 2, 7, 9, 10. These are the clotting factor which are dependent on vitamin K for its activation. So whenever vitamin K is deficient, all of this factor production will be decreased, activation will be decreased. If you look at all of this factor, shortest half-life is for this factor 7. The half-life of this factor is only 5 hours approximately, shortest half-life. So whenever vitamin K is deficient in your body, which factor is going to be reduced in your blood first? Factor 7. Factor 7 is which pathway? Extrinsic pathway. So what test is going to be prolonged? PT is going to be prolonged. But when your vitamin K deficiency is chronic, all of this factor concentration in your blood is going to be decreased. And here you can find out factor 9, 10, they are the factor of common pathway. Common pathway factor deficiency means prolongation of both PT and APTT will occur. So that's why it is written here that vitamin K deficiency, which is the late vitamin K deficiency, chronic vitamin K deficiency. But if there is a vitamin K deficiency in early stage, it will only prolong PT. But in late stage, in chronic stage, both PT and APTT will be prolonged. Not only that, any factor which is pathway of common pathway, they can also prolong your PT and APTT both. Now, when your APTT is only prolonged, I have to think of intrinsic factor deficiency. If there is deficiency of factor 8 and 9, significant bleeding will happen, and that is nothing but hemophilia. But if there is deficiency of factor 11 or factor 12 also, clinical bleeding is not very prominent or no clinical bleeding at all, because these factors are redundant. They are not very important in the coagulation system because of the amplifying nature, because of the positive feedback system generated by thrombin, right? So this way you remember the coagulation pathway, how it is activated and how it is working in your body. Lastly, you look at this table. Obviously, you are not going to remember all of this information, but here I have kept this just to look at the half-life of different clotting factor. If you look at closely, you will find out that the factor 13, that is a fibrin stabilizing agent, it has the maximum half-life, and the shortest half-life is here. Already we have told you the factor 7 that has the shortest, the minimum half-life among all of these clotting factors. Others are also given, but it is very difficult to remember. So this is all about your hemostasis or blood coagulation.